In this video, we will continue our discussion about this AOH1996. In the previous video, we have understood the basic understanding that AOH1996 will upregulate this transcription replication conflict in the tumor cells. So here we do not see any kind of proof, but in the today's figure, we will see some evidence of this AOH199 specificity and how it target and what are the results of this AOH1996. So in order to check the effect of this AOH196, they took a pancreatic cell lines in which when the doxycycline is introduced, then it will cause KRAS mutations, and they check the effect of this AOH in that conditions. So for the control, and they use only DMSO, and they see then when there is no doxycycline, we do not see the KRAS G12D mutations, but when there is doxycycline, we see KRAS G12D mutations. And this gamma H2X is the indications for the DNA damage. So once the DNA damage occurs, this gamma H2X gets phosphorylated, and we can see its signal in the western blot. But in the case of DMSO, when there is no KRAS mutations, we do not see any when there is no KRAS mutations, we do not see gamma H2X, and when there is a KRAS mutations but there is no AOH, we do not see any gamma H2X, indicating that in the normal pancreatic condition, in normal mutated conditions also, there is not much DNA damage occurs. But when this AOH is introduced for 24 hours, this 10 nanomolar concentrations, without doxycycline, we do not see gamma H2X or very minimum gamma H2X signals were there, indicating that this AOH does not target the healthy cells. But when they use this doxycycline that cause KRAS G12D mutations, we can see this gamma H2X formations, indicating that this AOH will target the cells which has this KRAS G12D mutations. And next, what they did is time dependent cell growth assay, where they check the cell growth with this AOH treatment at and what they are able to find out that when the doxycycline is introduced, they see a continuous decrease in the growth. They see a significant decrease in the growth after a specific time period. And when the doxycycline is not introduced but AOH is introduced, they see a normal growth in the cells. That can be seen in the figure B. And thereafter, for the orthogonal validation of this, they use a ICRAS cell lines, ICRAS murine based cell lines, in which when the doxycycline is introduced, the KRAS mutations are there. So they see that when they don't introduce doxycycline, the viability of cells does not go down significantly, but when they introduce doxycycline, the viability of the cells goes down. And this further substantiates their previous finding that AOH is targeted specifically to the oncogenes, the tumors or the tumor cells which has this KRAS G12D mutations. Next, they check some of the other pancreatic cell lines that has these mutations. So some of them human pancreatic cell lines and the murine pancreatic cell lines, and they are able to see a decrease in the viability in all the cell lines. And one more thing interesting they find out in this case is that BXP3 does not have these KRAS mutations, but they have other mutations similar to the KRAS mutations, indicating that their AOH1996 is not specific to the KRAS mutations, but to the oncogenes that triggers the tumors. Now they wanted to check the effect on the high metastasis and low metastasis, whether the high metastasis cells get whether the high metastatic cells are more affected by AOH196, or both of them has the similar effect, that is high metastasis cells and low metastasis cells. And when they check the viability assays in the high metastasis cells and the low metastasis cells, they see results that is about IC50, that is shown in the, they see a difference in the IC50 in the high metastatic cells and the low metastatic cells. And those results are shown in the panel lab, where we can see that low metastatic cells has high IC50 compared to the high metastatic cells, indicating that this AOH196 targets more aggressively to the high metastatic cell lines compared to the low metastatic cell lines. So this is what as the p-value is less than 0.05. So this in summary, this figure indicates that is AOH1996 can target oncogene, can target or affect the oncogene dependent growth and results in the growth inhibitions. So that's all for this video. If you need to understand more about their experiments in detail, you can go through the reference that has been provided in the description. Thank you for your time.